So let me know when you start recording. Yeah, which side, but good. I just need to.
Ready?
Yep.
Hello everyone. So welcome and thank you for giving me the chance to talk. My name is Kidger. I'm one of the product and application specialists at Zeiss. My main responsibility is in automation and software, which is why I'm here, because the Cell Discoverer is primarily the main microscope that I use because of its high automation, free functions, and the fact that we pretty much have to analyze every image that comes out. So this is a Cell Discoverer. I'm not really going to go into too much detail about the talks because about the hardware itself, because Tom has already discussed and mentioned a lot of these things. But fundamentally, the Cell Discoverer is what we would call an automated microscope. So it's a little bit one step on from what we call a motorized microscope, and that it was aimed primarily to do a little bit of the thinking for you. So you don't need to be a microscopist to use this microscope. The automated features are there to be able to take the information that you're using and automatically set the microscope up perfectly, so you don't have to worry about things like spherical aberration or any kind of artifacts because you're using glass bottom plates or plastic bottom plates. All of the correction colors will optimize themselves for that. But what it leads to quite nicely is that because it is easy to use, we generate lots and lots of images with it, and a lot of those images can be used, can be taken with the Airy scan, so at super resolution. So the resulting output from the Cell Discoverer can be quite huge amounts of data. And if we look over the years, I remember back when I first started with in this kind of job back in 1998 or so, I remember saying to one of my colleagues, I've got a 16 megabyte hard drive and I can hold all my work on it and all my images on it. But over the years, if you look at my desk now, it's like a mini server room. I think I have 20 terabytes in my desk at the moment, and I'm forever getting data sets and hard drives emailed to sent to me through post or emailed to me. And what's happened over the years is our images have gone from being, a few hundred megabytes with things like the lattice slide sheet, or if we're screening, as some of my customers do, 24 well-played organoid screening with Airy scan. We're generating single files that are close to a terabyte in size. And if you're working with images that are a terabyte in size, unfortunately, what hasn't increased at the same rate as image acquisition is the amount of RAM that you have inside your PC. And this difference between the two can make image processing and image analysis quite difficult. Traditionally, if you had a 1 gigabyte image, you could quite easily process and analyze that with a laptop running 4 gigs of RAM without any problems and no lag in your system. But if you've got a one terabyte image, it's very hard to find a computer that's got 4 terabytes of RAM in it. So what I'm going to talk about is how we can address some of these big data problems with the software that Zeiss provide. Next slide. So Zeiss has a software family now. We have our Zen software, which is used to basically control all of our microscopes, whether that be Zen Blue, Zen Black, or Zen Core for material science. Zen will always get involved in the image acquisition and the building of experiments. They can do pre-processing and post-processing and image analysis as well. But a few years ago, we acquired a company called Rebus, who specialize in three-dimensional big data analysis. And now we work closely with them. So when we're dealing with huge data sets, we tend to move away from the Zen acquisition software and into the Rebus family to deal with a larger high content, high throughput, especially with robot kind of analysis, the kind of data that comes through. So Zen itself, as I said, it is the automation software that we use to control all our microscopes. There's a lot of nice tools for it in there. It's very modular. We have things like smart acquisition, which is what Tom and Serge talked about with the guided acquisition workflows. Because it has its own inbuilt AI toolkit, so we can use AI for machine learning classification, machine learning segmentation, even deep learning denoising. So it's quite a powerful system in its own right. It has got image analysis, It has got bio apps inside there as well, so you can build your own image analysis. And it can do 3D measurements, but it doesn't really deal with the data as efficiently as our Rebus software. So if you're dealing with sort of the hundreds of gigabytes to terabyte data sets, then you're much better working with the Rebus platform for your image analysis inside there. Tom. Okay. So what I'm going to talk about are some of the tools that are found inside Zen to deal with this big data problem. And there's two approaches to it. One is you can build smart experiments. So the idea is that you try to mitigate how much data you capture. You only capture the things that really mean something to you, and you ignore the fields of view in your samples that don't need it. So that's going to reduce down the amount of data you collect. And the other approach we can have is dealing with software which is optimized to handle these big data sets. So we'll go back to the first one. So trying to create smart microscope experiments. So one of the tools that we have in Zen, it's on your system and on Sergio's, I think most in fact, all cell discoverers in the UK have got direct processing involved. And this is where we can link up our acquisition system with a processing system. So configuration 2 that we have on here is what we normally set the systems up with on cell discoverers. The idea is that our acquisition computer is working away, acquiring new images, and at the same time, we can do this parallelization of the processing. So we can send across, as soon as the first Z stack of the very first image and the very first position of your data has been acquired, that stack is sent across the second computer, and that one there will be running whatever processing steps that we're using. So it could be things like area scan processing. So these are the processing functions that we've added into direct processing. Some of these are maybe you don't have, Serge, because these are, every time we're releasing a Zen, we seem to be adding more and more processing steps into the direct processing, because it's becoming essential that we can utilize this parallel processing on our data. We imagine screening a 96 well plate with 4 colors, multi-position in each well in Z. And then you want to do area scan processing at the end, or you want to deconvolve it if it's a wide field experiment. At the end, you may be taking a 400 gigabyte file and pressing the deconvolve button or the area scan processing button, and then that will just run on and on for a few hours. If you can take small packets as you're acquiring and process them on the fly on the second computer, then once you finish your main acquisition experiment, it may only be a few minutes before the second PC has finished doing all of the processing, and you can be ready to take your data and do your analysis at that step. So I would advise anyone that's building any experiments with cell discoverers to fully take advantage of the module direct processing. Things like area scan processing, the LSM Plus and deconvolution are really, really useful. Extended depth of field, again, if you're taking big Z stacks and you really want to minimize file size to the max projections, we can automatically build this into the pipeline. And the idea with the direct processing is that we can daisy chain the processing events. You would just add a processing step into the blocks, it would send it off to the second PC, and it would just daisy chain these in together. So by the time your acquisition's finished, you don't have your raw data anymore, you have your process data that you can do your analysis on with really minimal delay. The next thing I want to talk about, and Tom and Serge mentioned it, is the guided acquisition. This approach, the idea is that, you know, instead of capturing, maybe you need to use the area scan to see what you want to see, but maybe they're rare events and they're scattered across the cover slip or they're scattered across the well plate. The idea with guided acquisition is, instead of saying, this is what some of my customers do, they just pick a 5x5 tile and they scan the whole thing at area scan resolution on the off chance that one of the frames has got what they're looking for. And then the file that comes out of that is huge, and 90% of it is unwanted data. The idea with the guided acquisition is that you can really target what you want and only acquire that at area scan resolution or whatever resolution you need, and ignore the rest of the data, which is just being captured quickly on an overview scan. Just quickly click. So the idea is that, first of all, you pick ideally the lowest magnification that you can successfully use image analysis on to identify the objects of interest. This can be done out of the camera, and so we can scan really quickly at really low resolution, and maybe only take, for a 96 well plate, 4 tiles to capture one whole field of view, for instance. Or if it's in 3D 4-well plate, we can capture a whole field, a whole well with a single image. And if you can then use image analysis to identify the target that you want, you can then automatically switch to the water objective or whatever, add the water, turn the area scan on and acquire that. Just only those objects at area scan resolution. And again, you're speeding up acquisition and you're reducing the actual data file that you're acquiring. So at the end, we get a really nice high resolution area scan image of the organoid. And we haven't picked out any of them. The software has just automatically identified them and then changed the microscope and acquired only the organoids at interest rather than screening the whole field of view. What is really nice is that you can take this approach, this guided acquisition targeted approach, but also couple it together with direct processing. And this can be done in a number of different ways. We could acquire an overview image, apply direct processing, so maybe we need to do a max projection so we can count foci in the cells and then say, okay, if the cell has got X number of foci, we want to then continue and scan it. So if we're doing a Z stack, maybe it's difficult to do the 3D analysis on the foci within Zen. So we can use direct processing to automatically max project the images. Then we do the image analysis on the projected image, and then we use that information to tell the microscope exactly where to go to find these cells and only image the cells. So we can really mix and match this on-the-fly processing with this targeted acquisition to really reduce time and reduce data size and capture the data. Here's some examples. This was a funding for mitotic cells. So here we just set the image analysis up to find the dividing cells, and then we want to capture some really nice area scan images with them. You can just click through. Again, here the main interest was that Chris wanted to find these touching cells. And they were few and far between inside this application. So really, when we met them the first time, they were screening everything at high resolution. And now these low ones were done with I think a 20x wide field camera system, whereas the main acquisition one was done on the area scan. Last example here, this is a 35mm dish with organoids. This was a really simple one, where this wanted to screen, find all the organoids, and then do area scan processing. What you can see as well is that we've got the overview image here. So this is a combination of using guided acquisition and Zen Connect. Zen Connect allows us, it's a really good way of working with guided acquisition because it will automatically capture your low resolution scan. And then when you start doing your high resolution detail scans at the various points, it will automatically superimpose them into the right position on your low overview scan. Zen Connect itself has a project database, which will organize all of your images. So then when you want to then look at your data sets, all you need to do is open up your Connect project image here, and then you can just double click on any of the organoids, and it will automatically open up the file, the full 3D data sets of those files for you. So it's a great way of visualizing and organizing your data sets in an easy way. One of the most underused features of Zen, possibly because it involves a little bit of programming, is experiment feedback. It's a tool, you can use it a little bit like guided acquisition. It can do some things that you can't do with guided acquisition, but it is a little bit Python driven. So here, the idea was, again, a customer wanted to scan a well plate, but they're only interested in really counting, I can't remember exactly, 200 cells per well. Then they just wanted to move on to the next well. And some of the wells, they had loads of cells in them, they were highly confluent. Other wells were not so highly confluent. And again, their approach was just to scan the whole well on every one, because they knew that the non-confluent ones didn't have enough cells that account pretty much every one. So the acquisition time took a long time, and the amount of data they captured again in some of the highly confluent cells was far too much that they needed to use. The idea with experiment feedback, as I said, it requires it's actual tool inside Zen, so it's got a graphical user interface. If someone's already written the scripts for you, like maybe Maria or somebody, then you don't need to know any programming at all. But it is a little bit of a you need to set it up. You need to have some skills in programming. So the idea here was that we set up a scan where it would automatically count as we were doing the acquisition. So we'd go to well one, take an image, analyze the cells, count how many there were in there. If it was less than 200, it would take another image. If it was less than 200 still, it would take another image. Once you hit the 200 point, it would then just move to the next well automatically. And it would continue on all around the whole plate. So they got the 100, 200 cells per well, but the amount of data they captured was much, much less because they're using this sort of smart approach to acquisition. We have lots of examples already of experiment feedback where you can take the code and just modify it yourself. You don't need to start from scratch. Part of my job to help you build these things as well. But it's a tool that I try and encourage people to use a little bit more than they do. I think everyone gets scared when they click on the edit experiment feedback script, hope to see some little buttons and GUIs that they can use, but they just see a big white page. And then it's down to you to fill it in with code. So they were 3 different ways of I guess, taking a more smart approach to building experiments. The next approach to dealing with big data, what Zeiss took, was that we acquired a company which specialized in dealing with image processing of and image analysis of big data sets. And this is a company called Rebus. Some people may know it already. It used to be called Rebus Vision 4D, but since Zeiss rebranded this year, we now have a software called Rebus Pro, but it's exactly the same as Rebus Vision 4D. And we're using this now to really deal with all of our really complicated image analysis and take advantage of the fact that really you don't need to use supercomputers. I know Andrew's given a really great talk about high content computing, and you guys obviously are going to be very lucky to have such a resource in there, but not everybody can have access to the high content computing that Andrew's setting up. And utilizing a software which is optimized to take into account things like parallelization and blocking, using the pyramidal format to load up the plane of interest on demand, so you're not basically getting redundancy of resources. This is what this software is really built around, as well as having very advanced image processing and image analysis techniques. The idea of how it handles it, it creates its own proprietary file format, a .sas file. So we take our images from the 87, you drag and drop them into the Rebus Pro software, and it automatically gets converted into this SAS file format. And it effectively is limitless on the amount of imaging channels or time points that you can use. On my laptop, I routinely analyze 1 terabyte images, unfortunately. It isn't the quickest, but it doesn't fall over. Ideally, we're just setting up some servers in the Zeiss office where I'll be doing a lot of this processing on. But at the moment, I know I can press start on my computer on a Friday night, and it will run all weekend, but it won't crash. But eventually, I'll get data at the end of it, which is really great. But it's really optimized, as I said, to take into account all these computing techniques that Andrew mentioned with the blocking and the parallelization of. We try to parallelize as much of the image processing steps as we possibly can. We take advantage of GPUs. If you have GPUs, any of the processing that will be optimized for GPU processing, we take advantage of that as well. So the software itself is very modular, a little bit like Zen. I can't remember exactly what modules you have in Liverpool, but we certainly have the visualization analysis. So this allows us to do 3D image processing on 3D, 4D data sets. We can do image analysis. You can measure distances and relationships between objects. We can count things inside things, but with huge data sets. Here's some optional modules. So we have a neuron tracing tool inside there, which can be added on, tracking and lineage measurements as well. So we will deal with, as you can see here, hundreds of thousands of tracking measurements on the fly. The import of objects may be useful to the EM kind of team. This is if you already have segmented data sets already from maybe microscopy image browser somewhere where you've traced around manually, like you used to do in the old days, and building your segmentation data sets. You can still take those binary masks and drag and drop them into Rebus, and that will generate a 3D image already that you can then use to measure if you've already got segmented data sets. The last one is the volume fusion. This was a light sheet data not a Zeiss light sheet, another light sheet data, where the customer couldn't fit the whole organism into his light sheet. So he actually imaged this in 8 different parts. The limbs, the head were all imaged separately, and the volume fusion brought it together in some kind of 3D volume stitching kind of algorithm. So all these tools can be plugged in, but the main tool is the 3D analysis, visualization, and processing tool. Go back one step. And the final 2 icons at the bottom, we can do all this in batch. So you can, if you've got tens of well plates that you want to analyze in 3 dimensions, we do have a batch tool inside there, so you can load up lots of data sets. You can find the pipeline that you built your analysis on, and you can just execute them. And they will process away, but on the local PC with your setup. So it will tie up your PC for the next X number of hours or days, depending on how big your batch is. And the last icon relates to our deep learning tools. The Erebus Pro has a lot of deep learning tools. And we have inbuilt Salesforce tools inside there. You can import Stardust applications in there. We have our own deep learning local trainers. We can take deep learning models from the Erebus Cloud. You can even import your own deep learning models if you've scripted them yourselves, as long as they're in sort of an Onyx format, then you can import them in. So if you're coding yourself, then that's fine. Another part of the Erebus family is the Erebus Cloud. So the Erebus Cloud, some people may have used this in the past. It used to be called Appear. It was launched by Zeiss in 2018, sorry. And it is a online computational and image processing tool. The uptake, I guess, from 2018 to lockdown wasn't so great. And then we introduced the machine learning side of Erebus Cloud, where you can use this to easily train your deep learning models in the cloud, download those models and use them in Erebus or Zen, or if you like to live in the open source world, you can even take the models from here and plug them into Napari if you really want to do sort of open source analysis. But the nice thing about the Erebus Cloud is the way we've optimized the annotation and training steps. So we've added in a feature called partial annotation. So if you're building deep learning models, you do not need to annotate every single pixel of every single image to be able to give you the ground truth for your deep learning models. With this partial annotation, we can get away with maybe 50 or 100 annotations. So you don't have to spend hours and hours and hours training. So your training time, your annotation time is really minimized. We also introduced a lot of pre-trained weights into our model building as well. And the speed of our model building since we first launched it, maybe an average data set might have took something like 4 to 6 hours to train. Since we've optimized the algorithms, we're now training these things in maybe less than an hour or 2. So really speeded up the way of generating your AI models. But because it's on the cloud, you can upload a subsection of your data set, do your annotations on it, press train, then you can go off and your local PC isn't used up at the time, and you can do whatever you want while this is training. It will then send you an email to say your model is ready, then you can download it, and then you can use it wherever you want, in Zen, in Erebus, or in Napari if you choose to do it. Next, I want to talk about some of that you don't have a Liverpool, maybe you won't ever need it, after listening to Andrew's talk, is if you really need to upscale your analysis. So you built your Erebus Pro, you built all your pipelines and that to do your analysis. You've been using the batch processing tool to process them, but then there's 10 other people wanting to get on the system because somebody's just hogged up the resources for the next 10 hours. Erebus have a server-based tool called the Erebus Hub, and this can be installed on your local or cloud-based servers. And it basically allows you to upscale your analysis from a local PC into cluster processing or cloud-based processing. You don't generate or build your pipelines in there. You just take your existing pipelines from your library, from your Erebus Pro. You upload them to your Vision Hub interface. You then link that to your data set. So there's no duplicating of data. Gone are the days where you need to download a copy of your data set from the acquisition system, and you've got a copy, you share it with your PI or your collaborators, and there's 6 or 7 versions of this thing going around. The whole idea is that you generate one image, it's uploaded to the cloud, all your collaborators can log in from wherever they are and they can get access to the data. And we can take into account, we can do the parallel processing. So you would buy a number of what we call analysis workers. So if you bought 10 analysis workers, then we can process 10 data sets simultaneously. And this again is speeding up the acquisition, much like the high performance computing that Andrew mentioned earlier on. So this is how we would structure it. So it is basically stored on your server. Like as I said, it could be a local cloud cluster, or it could be in the cloud, like a Amazon Web Server, whatever. And the idea is that we upload all our data into a server. We build all our pipelines on the Rebus Pro, and they get sent up into a library on the server, on the Rebus Hub. And then from wherever you are in the institute, if you've been given access to log into the server, then you can log in, whether it be with your smartphone or with your internet browser or tablet. And then you can then find the pipeline that you want to use on your data set. You can find your data set, you can link them together, and then you can tell it to start processing away. You can also take advantage of the fact that the 3D engine or GPU 3D engine can be sitting on the cloud as well. So when you then want to look at your data, if it is on your tablet and it is a 1 terabyte 3D stack in time, you're not using the resources of your tablet to try and open that. You're using the resources of the 3D engines stuck back in our server here. The idea again is that it allows you to visualize your data. So here we have the 3D or the 3D engine, which is based in the web browser, where you can log in, you can browse your image data set, you can double click it and then just look at your data from anywhere. All the folders are user controlled. So again, it's down to the IT administrator to decide whether you want people to get access to the data or whether it's just you can see them only. So it's fully user managed, I would say. The idea is that we take all the data that we're processing and we can then do all of the result visualization online. So we don't need to even download the data to do our charting or generate our heat maps. We can do it all online. And then, as I said, anyone in the group or with collaborators, you can share links to the results that they can also just log in from anywhere, look to the data. And so we're trying to really eliminate this movement of duplication of large files all around the university by working on the one active document. As I mentioned earlier, that we do have this full user requirement. So when you create a folder, you can decide whether people can get access to it, or can read it. They can run jobs on those images, or they can just look at the images. It's all fully controlled by the administrator itself. And then it's all visualized and viewed through these browsers or tablets or whatever. So it is a commercial solution. It's just a software-based solution, so it's not the hardware. We don't provide any of the hardware. We just provide the Vision Hub software, which is installed on your servers, and it just controls all of this data analysis. One thing I should mention, one of the main differences between the Rebus family and the Zen family is that the Rebus is system agnostic. So we don't care whether your data sets come from Leica microscopes, Nikon microscopes, whether they're EM images or micro CT or LM images. We can put all of these images in. You don't need to convert them into another format and then put them in. You can just drag them in. They'll automatically get converted to the CIS format, and then you can work with them. So it is and always will be what we call a system agnostic software. Obviously, it's owned by Zeiss, but we are always going to keep this side of the company system agnostic, and we want to work with whatever microscopes anyone has. And whereas Zen fundamentally will only work with Zeiss images. That's the main difference between the two platforms. So in summary, Zeiss have tried to build together a software portfolio which allows for easy automation and easy scaling up of your data sets. We're working very, very hard to provide leading edge AI tools. Our AI infrastructure, whether it be in Rebus or in Zen, we call it an open ecosystem. So it allows us, we don't really care where the model has come from, whether it's come from somebody else, a data analyst who scripted themselves. As long as it's in Python, we provide a Python library which you can convert into a Zen format, and then you can just drag and drop your AI toolsets and use them so you can stay really cutting edge with our approach. The idea with the cloud is that you can access the data from anywhere. You don't have to be on the local systems. We can share and collaborate very easily. And yeah, the whole thing, we try to make as easy to learn as possible. And if it's not, it's part of my job to make sure that you can know how it's working. And with that, I will end my talk.
Thank you very much. If there are any questions, maybe we have them up.
Yeah.
We're running a bit behind. And last but not least, we've got 2 short talks about projects that have started using the CD7 already, and hopefully we'll show you as well what's possible and what we've achieved in the first months having the system. So I'd like to introduce Perry, who will give the first talk.
Perfect. Hi, my name is Kerry. I'm a clinical ophthalmologist and a first-year PhD student. I'm just going to give you a quick overview of what we've been using the CD7 for. I must say most of the work has been done by our postdoc, Dan Phillips, and then Marie has been instrumental in helping analyse the data. So our project is on developing inhibitors of the Pseudomonas aeruginosa exotoxin U. Exotoxin U is a phospholipase secreted by the type 3 secretion system, and we know in clinical outcomes of our clinical infections. That was good. I hope it'll last. So we know that exotoxin U is strongly linked to poor clinical outcomes. So patients that have corneal ulcers have a really poor outcome. The same with patients who have sepsis. So that's why I'm interested in it. A quick schematic at the bottom. So very cartoonized as such. The Pseudomonas attaches to human cells. It secretes the exotoxin U, which is this red molecule, by its type 3 secretion system. It binds with cofactors, localizes to the cell membrane, and then conducts its enzymatic cell lysis within 3 or 4 hours. Oh.
Oops, do that.
No, I'm good.
That's all right.
Okay, so we've been utilising the CD7 for high throughput screening. We've developed a modified scratch assay and used live cell imaging with live dead immunofluorescence. The EXO-U inhibitors, we're testing 3 different candidates that we've identified from our high throughput in vitro screening, and we're testing them at different concentrations and combinations against clinical isolates. We have a library of 660 isolates taken from microbial or keratitis or corneal infections, and the cell line we're using is a human corneal epithelial cell line. The left-hand side you can see, so we grew a subconvolute layer of HCT cells in 96 well plates. We add our inhibitors, different combinations, concentrations, infect with Pseudomonas. We've been testing different strains, and then we've been using live dead imaging. So the acquisition we'll be doing on the CD7, we take images from each well every hour for 17 hours. They're wide field images. We've been using the oblique channel and 2 fluorescence channels as well to pick up the live dead calcium, ethidium, hemodimer, and then using Zen Blue to do all the pre-processing and analysis, looking at live dead ratios and also morphological changes. So this is sort of the workflow which Maria's helped put together. So the raw images are taken. We then have been doing pre-processing on it to reduce the background fluorescence from those channels. So we first split or take the images, do the background subtraction on the fluorescence channels, and then fuse the images back together. And then we've been using Zen Blue to do the analysis as well. So some of it we've been using the pre-processing images. We convert it to Excel files for downstream downstream plotting and analysis using different softwares. But we've also been using the analysis tab on the ZenBlue to get some outputs from it. The sort of outputs that we get, so we get Excel files generated with perimeters, area, convexity, circularity, compactness of the cells. And the outputs look something like this. And then we have been getting graphs as well, which show live dead ratios on this side here, and then these graphs are the 96 wells that show the cell convexity over time. Oh,